Hello, I'm Monica Bravo.
I'm Jasmine Ha.
I'm Alexei Solonenko.
And I'm Austin Decker.
And our group did a presentation over the article titled 2' O methylation in mRNA disrupts tRNA decoding during translation elongation. Knowledge over post-transcriptional modifications of mRNA is increasing as the number of known post-transcriptional modifications increases. This study takes a look at how more recently 2' O methylation of nucleotides has been observed as a frequent modification in translated regions of human mRNA. Though little about the actual modification mechanism and function are still unknown, this paper took a look at how the elongation stage is disrupted by the 2' O methylation of the mRNA. It does this in several ways. The first is a steric interference with specific ribosomal monitoring bases in the small subunit. This interferes with interactions between these monitoring bases and cognate codon-anticodon helices. This steric interference also leads to the inhibition of downstream GTP hydrolysis by elongation factor EFTU. And since EFTU and GTP use the energy of hydrolysis to bring the next amino acyl tRNA into the A site, this initial steric interference ultimately leads to the excessive rejection of cognate aminoacylated tRNAs in initial selection and in the proofreading. And in analyzing the effects on translation, this study also looked at if antibiotics could affect this hiccup in the decoding machinery. So specifically, it looked at how the antibiotic paromomycin is used to observe alterations in protein biosynthesis that ultimately diminishes the disruption of 2' O methylation in mRNA. The hypothesis for this article is by discovering the regulatory and proofreading mechanisms behind 2' O methylation of mRNA, the dynamics of translation elongation can be better understood. Fluctuating rates of translation at specific mRNA regions may lead to different protein folding pathways, which greatly affects biological development. Here we just show a regular mRNA nucleoside and then a methylated 2' O methylated nucleoside.
In order to analyze the effect of 2' oxygen methylation on translation elongation, a SM-FRET method was used to assay in vitro translation. FRET involves distance-dependent transfer of energy of about 10 to 100 angstroms from a donor molecule to an acceptor molecule. It's a great way to observe molecular interactions. The fluorescence emission spectrum of the donor must overlap with the absorption spectrum of the acceptor in order to capture fluorescence during the experiment. For this system, an E. coli 30S ribosomal subunit was labeled with Cy3b, and the 50S subunit was labeled with BHQ2. This let them monitor conformational changes in the subunits for many codons and multiple cycles of translation elongation. A zero-mode waveguide, or ZMW instrument, was used to record around 10 minutes of translation by tethering a pre-initiation translation complex of the 30S subunit and biotinylated mRNA onto a Nutravidin chip surface. To make it easier to understand, here is a diagram of the Nutravidin chip. The mRNA contained lysine codons with one methylated base at each of the 3 different base positions. Then, translational factors, which were the 50S subunit and Cy5 tagged lysine tRNAs, linked to lysine tRNA synthetases, were introduced to the complex during data acquisition. The use of a second fluorophore allowed correlation of 2 independent signals during translation elongation, as well as observing possible tRNA sampling dynamics on the modified lysine codon. You can see the mRNA is bound to the chip right here, and this is the 30S subunit that is tagged. Here is the 50S subunit that also has a tag.
Okay, so there are 3 results to this paper, and I'm going to be going over the first 2. First result is that 2' O methylation delays tRNA accommodation to the modified codon. So figure 1C shows experimental trace data. The red indicates the fluorophore signal of the tRNA that will bind to the modified codon, and the green represents whether or not the ribosome is in the rotational state or not. And if you do this under different conditions, you get figure 1D. This shows the same experiment, but under different Mg2+ conditions, and this shows which part of the codon they methylated. This is the first nucleobase, this is the second nucleobase, or the third nucleobase modification. And you can see that from this data that there is an increase in the stall duration, that is the time in which the tRNA binds and a rotation event occurs, which indicates an accommodation of the tRNA. And this is not seen on a regular non-modified codon. Figure 2D is the same experiment, except they wanted to change the context of the codon, see whether or not that increases or decreases the stall duration, if there are discrepancies. And turns out that there are. Instead of AAA, which they use for figure 1C, they use codons UUC, CUC, and CCC, modifying the second nucleobase in each one, and they found discrepancies in the stall durations of each.
In order to get more quantitative data along with the SM fret, bulk kinetics methods were done to determine how a methylated codon affects tRNA accommodation and peptidyl transfer. This is a generic quench flow apparatus where you have two solutions that get mixed in a controlled manner and a quenching solution that stops the reaction at a desired time point. In this experiment, ternary complexes were mixed with 70S ribosomes. The ternary complex, which is circled in green below, is the EFTU, GTP, and tRNA with synthetase. In translation, the TC, ternary complex, is either rejected or accepted for GTP hydrolysis by EFTU. That's why GTP hydrolysis was tracked with a radioisotope. The ribosome concentration was the rate-limiting factor that helped determine the Kcat over Km value of GTP hydrolysis for both normal and methylated codons. Finally, proofreading was tracked in a similar fashion, but with the radioisotope on the FMET tRNA synthetase.
Okay, we're off to our second result, which is that 2'-O methylation impairs both initial and proofreading selection of the amino acyl tRNA. So this is figure 3, and as you can see, the data looks very similar to figure 1 and 2. Figure 3b is figure 3a, but as a contour plot, and this is sample trace data under one condition, and it's used to make the one of the bars of each of these bar graphs. So anyways, figure 3c showed that if you methylate a codon, that the sampling time, that is the time in which the tRNA binds to the A site, increases if you have a methylated codon. And then figure 3D shows that the average rotational lag time increases if you have a methylated codon. Now, the average rotational lag time with a non-methylated codon is very fast, faster than what their equipment could measure, which means it's faster than 100 milliseconds. But here you can see that sometimes it gets to 1.5 seconds for the rotational lag time. And figure 3E shows that if you methylate the A site codon, then this increases the number of attempted tRNA binding attempts. Okay, so figure 4, they wanted to test the kinetics of the initial selection of amino acyl tRNA. And so when amino acyl tRNA binds to A site, a GTP hydrolysis event with EFTU occurs, and that can be tracked with a quench flow technique shown here in figure 4A. And figure 4B and D is a ribosomal titration, and that is to determine the catalytic efficiency of GTP hydrolysis under conditions where the non-methylated codon is in the A site versus the methylated codon being in the A site. And what they found was if you have a methylated codon in the A site, then GTP hydrolysis is slowed by 300 times, and the actual results are down here in the bottom right. And finally, figure 5 wanted to see the effects of the proofreading selection of amino acyl tRNA. And so what they wanted to find what's called the proofreading factor, which is a quantitative number of the GTP hydrolysis events per one successful dipeptide formation event. And so they put the modified codon directly in front of the start codon UAG, and they put fluorophores on the tRNAs associated with the start codon and a fluorophore on the tRNA associated with the modified codon or not, or non-modified codon. And they were able to track the dipeptide formation. And from that, without getting into the math because I don't have the time, they're able to find the proofreading factor, which is this, what they call the amplification, the fast phase amplitude over the fast phase amplitude of a modified codon in the A site. And so the proofreading factors under physiological conditions, 5 millimolar Mg2 plus is 5.1, And under 15 millimolar magnesium concentration, the proofreading factor is 2. And an increase in the proofreading factor signifies increased tRNA rejection to the modified codon during proofreading. And the researchers also wanted to point out that 2' O-methylation did not induce substantial miscoding by other tRNA species.
For the next set of results, X-ray crystallography gave valuable structural data. The purification and crystallization of 30S ribosomal subunits was done on the species Thermis dermophilus. Mass spectrometry was used to verify identities of molecules before crystals were flash frozen in liquid nitrogen. Diffraction patterns, which is usually obtained by X-ray crystallography, were obtained, and the PyMO program read the data and produced the figures seen here.
The final result, 2-O-methylation was found to disturb interaction between the codon and anticodon helix and ribosomal monitoring bases. For this, the way they found it was they added aminoglycoside antibiotics to the system, shown by figure 6A. This shows that the rate of GTP hydrolysis is normal in the black line, and then with the 2 antibiotics are in the purple and the green line, and then the blue line is the methylated mRNA without any antibiotics. This is because the aminoglycoside antibiotics lead to missense errors in the monitoring bases, and paramomycin, which is one of the antibiotics, stabilizes the disordered state of the ribosome to allow for the bulky methylated side chain. This was seen through X-ray crystallography because when there is not an antibiotic in the system, there is no difference in the electron density, so there is no ability to differentiate between the ribosome and the methylated side chain. However, when paramomycin is added to the system, the ribosome is differentiated from the methylated side chain. This also can be seen through the KCAT in figure 6B, in that there is a difference between the normal, the methylated, and then the methylated with the 2 antibiotics. Finally, putting it all together, and with a previous paper they did on N6 methylation of adenosine, which they refer to as M6A, which is the bottom half of this diagram, they were able to put together a model where there are 2 proofreading steps after GTP hydrolysis. They take place in the two sections before and after the removal of EF2 GDP. This is important because it gives greater understanding of the way that mRNA is translated and regulated. For further questions, the big part is, why does eukaryotic mRNA need multiple mechanisms in place for the purpose of slowing down translation rates? And what would happen to an organism's lifespan if one of these regulatory pathways was non-functional? Finally, the researchers said that they hope that there's more studies on chemical modifications, because that could give more insight into the regulatory elements of the translation system. Thank you so much.